Hello everyone and welcome to this video presentation. Today we are going to dive into one of the most revolutionary breakthroughs in modern biotechnology and molecular identification, that is DNA fingerprinting. Within this video, we will explore the foundational scientific principles, the core analytical techniques, and the massive global applications that have transformed forensics, medicine, and agriculture. Whether you are a science student or just curious about how molecular biology shapes our world, this comprehensive overview will give you a clear understanding of how we can identify individuals at the molecular level. Let's get started. To truly understand DNA fingerprinting, we have to look back at its origin. The year was 1984, and a British geneticist named Sir Alex Jeffreys made a groundbreaking observation. He discovered that certain regions in our DNA contain hypervariable repeating sequences, which we call variable number tandem repeats or VNTRs. What made this discovery so vital was the realization that these specific sequences differ completely from one individual to another. This moment marked a critical turning point in biotechnology. Before this, classical identity methods like blood typing or physical fingerprinting were often unreliable or lacked definitive precision. Jeffrey's discovery introduced an unprecedented level of precision to human identification, permanently replacing old methods with a highly reliable molecular based system. Now, let's look at the underlying science that makes this possible. The human genome is incredibly vast, composed of billions of nucleotides. Interestingly, as human beings, about 99.9% of our DNA is completely identical to every other person on Earth. However, it is that remaining tiny fraction, just 0.1%, that makes all the difference. This fraction contains polymorphic genes that are completely unique to each individual. These variations are found mainly in our known coding sequences, the parts of our DNA that don't code for proteins, and they consist of repeating units. Crucially, these patterns are inherited strictly from our parents. Exactly 50% is maternal coming from the mother and 50% is paternal coming from the father. Because of this unique inheritance pattern, when scientists analyze multiple high variability locations or loci in the DNA and find a perfect match, that the mathematical probability of two unrelated people sharing that exact same pattern becomes astronomical. This is what gives DNA profiling its definitive power. How do scientists actually look at these patterns in the laboratory? There are three core analytical techniques you need to know. First, we have RFLB analysis, which stands for Restriction Fragment Length Polymorphism. This was the early standard methodology. In this process, specialized restriction enzymes act like molecular scissors to cut the DNA at the specific sequences, resulting in fragments of varying lengths. These fragments are then separated using a technique called gel electrophoresis, which sorts them by size to create distinct visible band patterns. Second, we have PCR amplification or polymerase chain reaction. PCR is a highly versatile cloning technique that solves a major problem, that is small sample size. It allows scientists to rapidly copy microscopic or highly degraded DNA samples. Thanks to PCR, even minute or ancient traces of biological material become viable for detailed molecular analysis. Finally, we have STR analysis or short tandem repeat analysis. This is the modern gold standard in genetic profiling. Instead of cutting the whole genome, it focuses on specific, localized short repeating units. By using highly automated fluorescence sequences, STR analysis can compare these regions rapidly and with incredible accuracy, making it the preferred choice for modern labs. Now we'll talk about applications in biotechnology and fields of application. Because these techniques are so precise, DNA profiling has expanded far beyond theoretical science. It is actively used around the globe to resolve critical biological, legal, and medical issues. Let's break down the four primary fields of application. The most famous application is forensics. DNA profiling allows forensic scientists to match biological evidence collected from a crime scene directly with the profile of a suspect. This serves an essential dual role in our legal system. It is a powerful tool for securing convictions against the guilty, but it is equally vital for exonerating the wrongfully accused. Next is paternity and identity. Because we inherit our DNA directly from our parents, analyzing familial relationships can clear up legal inheritance and immigration matters. It provides near perfect statistical certainty when confirming biological links. Third, we look at medicine and diagnosis. DNA profiling helps clinical scientists detect genetics anomalies at a very early stage, assesses a patient's long-term disease, and power custom diagnostic options tailored to an individual's specific genetic makeup. Lastly, it plays a major role in agriculture and biodiversity. Genetic tracking is used to trace animal and crop pedigrees to optimize selective breeding programs. Furthermore, it helps conservatives protect endangered species and allows authorities to track down and stop the illicit wildlife trade by identifying poached material. Next are benefits, ethics, and challenges. While the benefits of DNA fingerprinting are massive, we must also look at it with a balanced perspective, considering both its advantages and its ethical issues. Let's start with the major advantages. The first is unparalleled accuracy. It discovers two genetic matches with an incredibly minimal margin of error. Second is its ability to handle minimal sample yields. As we mentioned with PCR, this technology works flawlessly with microscopic damaged or even decades old biological samples. Together, these benefits have had a truly transformative impact, completely revolutionizing standard criminal investigative methodologies and modern medical diagnostics. However, we must also address the limitations and ethics. Contamination risks are a constant challenge. Improper sample collection or minor laboratory errors can introduce foreign DNA, leading to skewed or inaccurate results. Then there are serious privacy concerns because genetic data reveals our most personal health and identity details. Storing this data requires highly secure databases to prevent surveillance overreach and profiling abuses. Finally, the potential for misuse means that society needs strict modern regulatory frameworks to ensure there is no unauthorized access to private genetics. As we look to the future, molecular profiling and diagnostics will only continue to become faster, more accessible, and more deeply integrated into our daily healthcare and legal systems. That concludes my formal presentation on DNA fingerprinting and biotechnology. Thank you all so much for your time and attention today. I would love to hear your thoughts, so let's open up to the floor to any questions, comments, or discussion you might have. Don't forget to leave your thoughts in the comment section below. Thank you.